and you can be confirming and telling me if they are correct, okay? Are you together? We know the same information. So it's from what we did yesterday. These are the Punnett squares. So we're going to see, what we want to see is, number one, which alleles are there? Which one is dominant? Which one is recessive? Okay? We want to see which alleles are there and which one is dominant, which one is recessive. Then what gametes does the father have, and then what gametes does the mother have? Of course, the gametes that the father will have will depend on his genotype. We'll see what is the genotype of the father. Right, so let's see. This RH factor is a protein, blah, 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 blah. If the protein is present, then the individual is RH positive. The allele for the presence of RH factor is dominant and is represented by D. So D is going to be the dominant one, and this is presence of RH. Presence of RH factor. All right. Then the recessive allele is represented by small d. D is going to be the recessive one, which is absence of RH. Okay, thank you. If a mother is RH negative and the fetus is RH positive, there can be problems during pregnancy. When is the fetus RH positive? If the RH factor is present, which means this dominant one is present. If the mother is negative and the fetus is positive, there can be problem. A RH negative mother and a heterozygous positive father, a RH negative and a have a child. So this is the situation we've been given. Complete a Punnett square. This is an RH negative mother, right? Class, this mother is RH negative. What does it mean if this mother is RH negative? She doesn't have RH factor, this one. And how do you have RH factor? They said the presence of RH is represented by D and is dominant. So if the mother is negative, that means she only has these recessive. So the genotype of mom is small d, small d. Then the dad is heterozygous. Once they say heterozygous, it's like this. Big D, small d. So now we would say, what are the gametes of the mom? Small d and small d. For dad, D and the small one. Uppercase D and the lowercase d. Now, what do we then do? We then start crossing. They said complete the probability of the child being positive. Okay, so let's see. If this two meet, it's going to be uppercase D, lowercase d. For here, it will be this small one, this lowercase, and this lowercase. So it will be lowercase d and lowercase d. If uppercase D from the dad, this allele, this gamete from the dad, meets this egg from the mom, then what will the child have? The child will have upper D from dad and lower D from the mom. If the dad's gamete with lower D meets the mom's gamete with lower D, then it will be like this. So what is the probability? What is the probability? There is a 50% chance, right? That the child is RH positive. Are we together? Why? Because there is this one and there is this one. These two are showing you this child, when he has this uppercase D, it means the RH is present. And since it's dominant, that means the child is RH positive. Can you see that? So if the child ever gets this type of genotype, then this child is RH positive. Can you understand what is going on? Am I speaking gibberish or you can understand? Yes. Which one is why did you ask that question about to say, do you take the dominant and place them at the top? Where? Here, this one. The one you are questioning is this one. Okay. This one is because we have two. What we put in here is what we are told. Here we were told that in a RH negative mom. So the mom does not have this uppercase D. Or else should be positive. Because we've been told that RH is dominant and represented by uppercase D. If the mom is negative, she doesn't have it, that means she has to have lowercase and lowercase only. Are we together? The mom cannot be like this. Or else, if she's like this, then she is RH positive. The mom can't be like this as well. Remember, all individuals have two alleles. So we are trying to predict which two alleles does the mom have. And we are told that a RH negative mom and a heterozygous father. If the father is heterozygous, it means he has an uppercase D and a lowercase d. He has both of the alleles. That's heterozygous. That's being a heterozygote. That's being heterozygous. It means you have different alleles. So this is a heterozygous father, which means he has upper and lower. The mom is not heterozygous because she is negative. If she was heterozygous, she would be positive because she would be having the dominant one, and the dominant one is always expressed. It makes you positive. Are we together? So we have been told what the mom has and what the dad has. So this is father, Mr. Roderick. This is father. And we've been told the father is heterozygous. So that means he has upper D and lower D. Are we together, Roderick? Roderick, are we together? Tadisa, are you in the lesson now? You are late, huh? You are late for maths. For physics. You don't do what? You don't do, but isn't it fun? Are you enjoying it? So stay. Things can change, eh? We are doing it together. Let's do the second question. Here's the second question, students. Here's the second question. These are our type of questions. We don't run away from this. Let's find out what we have here. Hey, researcher, blah, 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 blah. The body, okay? We'll be told which letters to use for the alleles and stuff and the type of people. So here they are saying, first question, 2 heterozygous fruit flies with black bodies were bred together. So they said the bodies of fruit flies can be black or yellow. So let's say B for black, Y for yellow. A yellow body is a recessive, so Y will be small letter, B will be big, B is dominant, and Y is recessive. So 2 heterozygous fruit flies, that means it's BY crossing BY. They were heterozygous. They have the black and they have the yellow allele. Are we together? Are we together here? We know what heterozygous means, right? Yes. So now predict the phenotypes of the offspring and the phenotypic ratio for this cross. So predict the phenotypes. What do you always get when you breed 2 heterozygotes? BY is going to meet BY. What do you get all the time? Let's see. You are going to get B and B will meet, B and Y will meet here, then this B and this Y will meet in this one, then the Y and the Y will meet here. So what are the phenotypes? They are asking us the phenotypes. Are you seeing? Class? Hello? Yes, here they are asking. All right, great. Here they are asking us for phenotypes. Remember, you're not going to let me down. Phenotypes is what we see, right? Phenotypes is what we see. They are asking us for phenotypes to say, what do you see? Right. Can I ask you, when in these 3 here that I've shaded blue, what do you see? This is a black one, right? It will be black because black is the dominant, and whenever a dominant allele is there, it's seen, right? So you can see that we have 75% of this, or 3 out of 4 being black. And then we have 1 out of 4 being, being yellow. This is the only time you get yellow, when both is yellow. So that means you're going to say black as to yellow. Okay, the phenotypes, you get black and yellow phenotypes. And the ratio will be 3:1. Okay? Okay? Ratio of black to yellow is 3:1. Are we together? Can you see that? 3 black, 3 will be black, then 1 yellow. Can you see that? Students? Hello? Because 3 will be black and... Okay, can I ask you, what is the phenotype when you get this BB? This BB, what is the phenotype? What do you see? What type of a fruit fly is this? Obviously black, right? Because B is for B. What about when it's BY? What is the phenotype? It's black also, because black is dominant. You've been told that yellow body is recessive feature. So that means obviously black will be shown when it's there with yellow. So that means we are going to see a black fruit fly, and here another black fruit fly. So it's 3 black, 1 yellow. So don't make a mistake here. Now let's do the next question. Let's do the next question, please. All right, let's do the next question. Now, what is it about? Let's go. Here it says, draw a genetic diagram to explain the results of this cross. Which cross? In another fruit fly breeding experiments, researchers counted 124 offspring with black bodies and 121 offspring with yellow bodies. Draw a genetic diagram to explain the results of this cross. Use the letter B to represent the allele for black body, and B to represent the allele for yellow, small b to represent the allele for yellow body. Can you see this? We have 124 as to 121, 124 as to 121, the ratio, right? Class, can you hear me? Yes, we have 124. We have 124 as to 121 ratio. And now they are saying to us, predict what did the parents have? What are the genotype? So this, let me explain something. I think I need a board here. I need a board. Right. These are ratios that you must know. Let's talk about them, because let's just write a heading. Somebody write a heading to say ratios to know for genetics. Can you write it? Ratios to know for genetics. Okay? Ratios that you want to know, that you gotta need to know for. Right. I'm not going to give you their names. They have names, okay? But here we'll be saying, I want you to say, it is you have a 12 to 3 to 1, 1 to 2 to 1. You have a 9 to 7. Put these things down, and then we're going to check which one it is. Are you writing? You see, there are words that they give you, which are words like epistasis, blah, 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 blah. So here they would have said to you, the key word that we had here from this question that we are doing was, they said, let me go back to it so that you can say, they said, we have an experiment and researchers counted 124 offspring with black bodies, right? And black, they said black was dominant. So dominant here had produced 124. And they said 121. So that's either, remember dominant is capital, right? So we have the dominant fruit flies, which had the dominant allele, and they were 124. Then we have 121 offspring with yellow bodies, which means they had no dominant allele in them. They just had the yellow. So which ratio is this? What would it help? How can we predict the genotypes of the parents and the phenotypes from this information which says that, right? We have 124. That's the key thing, because this is like they have told you. It's like they have told you what genotype there is, but they have told you using ratios. Okay? 124 is to 121. I want you to know that. Let me go back to the types of crosses we did yesterday. Yesterday we had a situation. We did how many? In the first part, in the first section of part 2, where we were doing crosses, genetic crosses, we first had, was it homozygous and homozygous? Can you remind me? Yesterday when we were doing our crosses, they were important crosses. I did 3. Let me put them in so make people see here. We gave each other a situation. I wrote the notes. Let me go instead of rewriting them. And in this one, look at this. Is this genetic for Form 4s or for Form 2s? Let me see. Try to see it with me here. No, this is not for Form 4s. This is checkpoint stage 9. This is the Form 4s one. Right, look at this. That's one of the most important things that I was supposed to show you yesterday. Can you tell me, please, class, when we have a genetic cross of 2 homozygous, what did we get here? What was our output? 100% was heterozygous, right? Hello? Guys, can you see that yesterday when we did this, 100% was heterozygous? When we crossed, can you see? When we crossed 2 homozygous parents that were different. All the offspring were heterozygous. Okay. What about here? When we crossed 2 heterozygous parents, what did we get? A 1:3 ratio, right? Okay. What about when we crossed here? This is the most important one. This is the most important one. What about when we crossed a heterozygous phenotype, a genotype, and a homozygous genotype? What did we get? Do you see that we got a ratio of 1:1? Look here, class. Just look at this cross. This is the 3rd cross. When you cross heterozygous with homozygous, you get 2 heterozygous, 2 homozygous. Is it not? So if we say the ratio is 2:2, it will be simplified. All ratios can be simplified. If it's a ratio, you can simplify it. It becomes 1:1, right? So tell me the ratios we've seen. Which ratios? Let's do the basic ones that you need to know for IG. This one, this is 100%. When you cross two homogeneous, you get 100% heterogeneous. Which means 100% will have the dominant allele. All the kids. Okay? All the kids will be, if this is what we had done, all kids were going to come out being black fruit flies. If we had taken a black fruit fly that is completely black, purebred black, homogeneous black, and we take a yellow fruit fly, then all the kids would have been black because all of them would be heterozygous. So if you are told that all the offspring were black fruit flies, then you know what parents you used. You used homozygous parents. If you have a 1:3 ratio, then it means you had two heterozygous parents. But if you have a 1:1 ratio, then it means you had a heterozygous and a homozygous recessive. Homozygous here, this parent has the same type of allele. So if I go back to this question, and I look at it again, I'll see that we have 124:121. This is almost 1:1. It's approximately 1:1. 124:121. This is 1:1. It's almost a 1:1 ratio, this one. So we can tell that, what did we cross here? Tell me the parental genotypes. We crossed a heterozygous parent with a homozygous parent. Is it not? If we cross, let me say, if we cross two homozygous parents, two homozygotes, our ratios, we know what we'll get here. We said we got 100% be heterozygous. This is what you get. That's case 1. Case 2, if you cross two heterozygotes, then you get the situation which we got in part A. You get heterozygous as to homozygous, but it will be a 3:1. Okay? Are you seeing it? Okay, if we are doing phenotype ratio, not genotype. If we are doing genotype ratio, it would be like this: BB. We have BB as to BB as to BB. It would be 1:2:1. But this is what you get. But for phenotypic ratio, it means what you see with the eyes. It will be 3 black as to 1 yellow. So this is black as to yellow, which is what you get, which is, I think, what we wrote here. Black as to yellow is 3:1, because they want the phenotypic ratio, what you see with the eyes. So let me explain, because I know that somebody is asking questions right now, and I can't pretend like I don't know that. I know that. Let me just do it one more time, because this thing called ratio. So are you saying we should know ratios before the thing? Yes. Is that what you're saying, sir? Yes. We should know ratios before the exam? Yes. Ratios for what? For the 3 types of crosses that you're going to do first. These 3. This one, it's good for you to know. And then it's good for you to know this. So you're saying we should know ratios? Yes. What are the types of ratios? Because we seem to be talking about 2 types of ratios. Say, yes, there are 2 types of ratios. There are 2 types of ratios, sir? Yes. What are the 2 types of ratios? There's what we call genotypic ratio. which does not talk about what you see with your eyes. It talks about the alleles themselves. Genotypic ratio talks about the alleles themselves. Here we see what are the alleles. I can go back to the table. You will see that you had one when we crossed two heterozygous parents. We got one that was homozygous dominant. We got two that were heterozygous. And we got one that was homozygous recessive. These were the 4 things we got. Now, the genotypic ratio for this is 1:3:1 in this order. But when we say phenotypic ratio, we are saying what we see. It has nothing to do with what they had. It has everything to do with what we see. We see that this is a black fruit fly. This is a black fruit fly. These are yellow. This is a yellow one. These are also black. The one that are heterozygous. Heterozygous, when you see them, they are black because the dominant one is there. And the dominant one is the black one. Because we have been told, we are not guessing. They will tell you which one is dominant. So the dominant one is the black one. So if it's there, it's going to be in the phenotype. So we have 3 black, 2 are heterozygous. 1 is homozygous, but still they are all black. So if it's phenotypic ratio, it's 3:1 for black as to yellow. Do we have a question here? Anyone with a question? All right. So we must know it, yes. Then we must know it. Then what is the other one you must know? Is the 1:1 one, which is the one they asked in part B. You should know it, which is the third one. When you cross a heterozygous parent with a homozygous recessive parent. It's like you're doing a test cross. What do you know about this offspring, this time of? What you know is that you are going to get 2. Well, let's talk about it. You get 2 being like this and 2 being like this. So you're going to get 1:1 because you get 2 being like this and 2 being like this, which you will reduce to 1:1 instead of to 2:2. So this is the genotypic ratio and also the phenotypic ratio. For phenotype, you would say you have 1:1 for black as to yellow. Are we together? Are we together? Yes. So let's come here. So don't, there are only, come on now, students, you're not going to let me down. We've gone through the hard part. You know that if you have a 3:1 phenotypic ratio, you know what the parents were. These are the parents. They were heterozygotes. If you get 1:1, these are the parents, so they are checking if you know that. If the ratio is 1:1, these are the parents you crossed. Yes. 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 No. Yes. The only possible combination, the only answer you always get is heterozygotes. If both parents are homozygous like this and one is dominant, the other one is recessive, you will always get, all the kids will be heterozygotes. For 3:1 is this. You take the heterozygotes, you cross them as parents. This is what you get. Let's do the Punnett squares. Let's do the Punnett squares quick for you to believe. Do you agree? Let's cross. Because this one meets this one, you get big B, small b. Can you? Then this big B meets this small b, you get it here as well. Then this big B meets this small b, you get the answer here. Big B, small b. This big B meets this small b, you get it. So all of them are heterozygous. You see that? You see it? So now we got this ratio of the offspring. There were 124s to 121. That's a 1:1. This is a okay, you need to watch that video. You clearly missed it. You were clearly, I can tell you are not there. Phenotype is what you see. I'll explain to you. Okay, you check me. So here, what are the parents? The parents are class. Roderick, can you agree with me on one thing so far, Roderick? Right? Are you agreeing with me? Can we agree that on one thing? That when you get 124 offspring with black bodies and 121 offspring with yellow bodies, this is a 1:1. Can you agree with me? That 124 and 121, it's almost the same number, right? So that means this is 1:1, right? Because experiments are never 100% accurate. But the fact that you got almost the same number, that means it's a 1:1. So now, which one gives you 1:1 on these three? It's this one, right? This one. So do it. So put that. So there's nothing to wait for. So just know those three and you'll be fine, class. Everybody. Shalom, can you hear me? Shalom. Shalom. Why can't you respond today? Tadisa, can you see? So we all agree that the genotypes, sorry, not the phenotypes, the genotypes of the parents were these ones. These ones. Anita, is it not? Do you agree? Good. So now, what about their phenotypes now? What would be their phenotypes? This is where some people, what would be their phenotypes? This is black body. This is black body because black body is there. Okay? And then this is yellow body. This is yellow body because there is no black body there. There are only two recessive for yellow body. So, yes, this is what you see. Now, let's put the gametes. It will be big B and small b. Here it is small b and small b. Now, they say draw a genetic diagram to explain. We can do it in no time. Let's do the genetic diagram. Then you put one line up, which passes these three. You first put 3 lines, eh? You're seeing me. Let me do it nicely. Line number 1. Line number 2. Line number 3, which you connect nicely. Okay. Then you put line 2. Okay, a little bit far away like this. Then you put line 3. Yeah. Then here you can say gametes. What are the gametes? we take these ones, we put them here. It will be big B, small b. Then here the other gametes, small b and small b. Then we do the cross. Let's put the cross. Big B meets small b, you get big B, small b. Big B and small b meet, you get big B, small b. Small b here meets with this small b here, you get small b, small b. Small b here meets with this small b here, you get small b, small b. So what are the offspring genotypes? What are the genotypes of the offspring? We have big B, small b, and we have small b and small b. Okay, the genotypes means the alleles. It's big B and small b. This is what? Then we have small b and small b. Expected ratio of black S to yellow, 1 S to 1. 1 black S to 1 yellow. You can write it like that. Do you see how they've written it? 1 black S to 1 yellow. So you can even write that here on the phenotypic ratio here. Black S to yellow, you can say as well 3 S to 1. You can say 3 black S to 1 yellow. Can you see this? Everybody, you saw this, right? Okay. I'm gonna give you questions to try to do from here, related to the ones we've done so far already, because it's time up. And then I want you to do them. Finish the maths. Come on, people. And I want you to do this one as well. The assignment I'm putting on these diagrams related to the 2 questions we've done. You can go back and refer to this and then do and then write. And I'll be marking. I want to see how far you can do. This is a free 5 marks. We're going to be enjoying this 5 marks. We are going to enjoy it. This is a nice, if we get this type of question, we should be ready for it. For today, what I added, just make sure you remember these ratios, please. Please don't forget these ratios. Important things, we should have an ability to see important things. And we should make sure we don't forget those important things once we get them. So here is one important thing, which is ratios. Ratios that must be known. Genotypic ratios. I've done them here, okay? 1 is to 1 when you cross this, a heterozygous and a homozygous recessive. You get a 1 is to 1. Then you get, there's no ratio here because all the offspring are heterozygous. All 4. So you get 4 is to 0. all the if you are going to cross homogeneous and homogeneous, you always get. But if you cross heterozygous and heterozygous, so we crossed heterozygous and homozygous, homozygous and homozygous, heterozygous and heterozygous. So these are the 3 we crossed. And here you get a 3 S to 1. Phenotypic ratio, genotypic ratio is 1 BB S to 2 BB, B small b, then S to 1 B, small b, small b. You should know this. We'll do more about it tomorrow. Of course we will, just to polish up and make sure we are not rusty. All right? Are we together? Let me just see. Anyone with a question? All right. Well done. I was just making sure that I have everybody written down. That's good. I have the register now. Well done, everybody. Well done. You attended. You see, attending is easy. Keep the things. I'm going to give you an assignment on both things we have done, and we continue again tomorrow.